You see yourself as a kind of explorer, and if you were using that metaphor for your PhD students and giving them advice on how to successfully explore, what would you tell them? What tips would you pass on to a new PhD student to make their life interesting and productive?
That's a difficult question. I always like to start from something that actually I don't quite formulate the problem in that sort of way. I mean,
well, ask yourself the question you'd like to be asked about it.
Yes.
Now, all this can be cut, so don't worry about it.
Yes.
I mean, I put together two questions. One is what advice you give to young researchers when they start their research, but perhaps unfortunately linked it to your idea of your own desire to explore and find out new things. So you can separate two questions off.
I had the great privilege when I started my research career of being in an area that was, when I started, was non-existent. It rapidly became rather hot. Everybody was doing it, and I got out. And the next thing that I did was, I got a junior research fellowship at Christ's, and my plan was to go and do a postdoc in America, and then come back and take my fellowship to molecular biology to Max Perutz. And I spent a year doing NMR proteins. It seemed to me the future of chemistry was in biology. Then my supervisor got a job somewhere else, and a vacancy came up in the chemistry department in Cambridge, and I got the job, so I never went to molecular biology. But I've always been inspired by biology. So everything I've done is either to use chemical techniques to solve some problems in biology, or use biology as an inspiration to do things in chemistry. But everything I've done seems to be by accident. So I had a phone call once from Adam Brooks from somebody who said, I've got a problem with certain kind of how drugs work, how certain kind of drugs work, can you help me? And that led to a whole project. I had a phone call from Max Perutz in the late 1970s saying, I have a problem, I'm told you're the person who can solve it for me, and that led to a very nice collaboration paper. And so I haven't ever actually initiated anything. Problems come to me, and then we make unexpected discoveries, and then I say, oh, I wonder if that means, I wonder if we can do this, I wonder if we can do that. And most of what I've done that's been successful have been things that other people have said can't possibly work. But when I've done it, I haven't known that other people have said it can't possibly work, and they have worked. So my advice is always to find an area that looks different from anything that anybody else is doing. There is no point, if there are 100 research groups out there trying to synthesize a particular molecule, there's no point in being the 101st group. Complete waste of time. You won't make any impact. And I don't think there's any point in doing anything, in doing a piece of research that isn't going to have some kind of impact. I mean, partly you do it for your own satisfaction. You know, I wonder how this works. But actually there's no point in doing it unless you can publish it and someone else is going to look at it and say, that solves the problem and that tells me how to do this. So I do think you need to have the feeling that what you're going to do is going to be noticed and it's going to make some impact. That's not the same as saying I'm doing applied research. Almost nothing that I've done has been applied in the sense of being picked up by a pharmaceutical company and used to create new drugs or new paints or whatever. One or two things have, but that's never been my motivation. I like to understand things. I like to help the world understand things. I like to develop techniques that solve problems in a way that people haven't been able to solve them before, then other people can use them. And once I've done it, I then move on to something else. When I was a PhD student, I sat next to an visiting American professor who was on sabbatical in Churchill, and he said, well, I've been in this field for 15 years, and now I'm writing a book as that's my gift to the field, then I'm moving on to something new. And that's something I'd like to do as well. Because I worked in NMR for a long time. I wrote a book, I did a second edition, but I've done no new NMR since then. I've gone to other things. And when I stopped taking PhD students and postdocs, the plan is to write another book that summarises the last 30 years. What I think I've learnt.
I mean, there's so many questions, but one is obviously what you did next. I mean, there's 30 years that you...
What did I do next? So as I say, my plan was to move from chemistry into molecular biology through the Junior Research Fellowship at Christ's. But a Junior Research Fellowship versus a proper university post, I had the opportunity of the post in chemistry. So I went to chemistry in 1973 as a demonstrator, assistant lecturer. By the time I got there, my supervisor had decided not to leave after all. So there were two of us in the department who knew about NMR and mass spec. And I was under huge pressure then from the professors to leave because they felt they didn't need two people in the department who knew. But I got tenure and I'm still here 30 odd years later. So my first project was I had thought about a way of doing NMR spectroscopy of big biological molecules that I thought was rather clever. And I got a grant for it. It turned out to be completely impractical on 1970s equipment. It's actually now a technique that is around. Other people have, as it were, rediscovered it, but it was not possible in the 1970s. But when I came back to Cambridge to this demonstratorship, I was sharing an office with my predecessor who did not get tenure, which was very uncomfortable. He was a nice man. We disagreed about music, but he was a nice man. And I did some experiments with him, published a couple of papers, and that led me to... he had discovered a reaction that he didn't understand. I said, let's use NMR to look at your reaction and see if we can find what's going on. And all the lines in the spectrum went to funny shape, and we didn't understand what was going on at first, but then we did understand what was going on. And we realised what was happening was that there were electrons hopping from one molecule to another in the solution, and the electrons hopping led to the NMR lines broadening. And the NMR lines broadening tell you basically how long the electron is spending on which piece of the molecule. And that seems to me rather an interesting thing. And just at that time, in 1974, it was becoming clear what the role of chlorophyll, the green pigment in green plants, what the role of chlorophyll was in photosynthesis, in capturing sunlight and turning it into chemical energy. And one of the things that happens is the chlorophyll absorbs sunlight and it gives one electron away, and that electron drives the whole of biological machinery, and that leaves chlorophyll with one electron missing. And what nobody knew was, where is the one electron in the chlorophyll? Well, we had just discovered, or rediscovered, this NMR technique. NMR is a technique invented by physicists, and it's full of the physics literature is full of techniques for doing things with NMR that chemists and biologists don't know about. And we rediscovered one of these by accident with my colleague trying to look at his reaction, and realised this technique could tell us where the odd electron was missing from the chlorophyll. So my first PhD student started in October 1974. We bought a packet of spinach seeds. The botanical gardens grew 150 row of spinach for us. He isolated chlorophyll from the spinach, and we did NMR experiments, and we found out where the odd electron was on the chlorophyll, which was quite interesting. And then that led me to ask myself, is it possible to make molecules which mimic the behaviour of chlorophyll in photosynthesis? In other words, absorb light, get rid of an electron, and then take an electron from somewhere else, and then generate basically chemical power. I mean, the whole of biology is driven by sunlight captured by biology, and that sunlight being used to separate electron from from the chlorophyll. And if you like, you can think of it this way, that electron finding its way back home drives the whole of biology. And so the question was, in 1976, 1977, could you build molecules that behave like chlorophyll in photosynthesis? You shine a light on them, an electron jumps somewhere and eventually finds its way back, but in finding its way back does some mechanical work. So I set my third PhD student onto making molecules like that. The third and the fourth and the fifth worked on that. We never made molecular machines of the kind that I described, but nevertheless we did discover some interesting things about how electrons move from one molecule to another. And we did that for several years. It all actually got too difficult. I realised that by then, by the mid-80s, there were 20 research groups around the world, all making artificial photosynthetic systems. And what they were discovering about their own photosynthetic systems didn't bear any relation to anything else. There was no generality. Everyone was kind of digging their own little hole, and there was no communication. So, using the same kind of molecular building blocks, we switched to making model enzymes which could bind molecules inside their cavities and then do chemistry inside cavities. And that kept us happy for another 10 years. Along the way, I had some wonderful PhD students, including one who is my first PhD student to be an FRS, Chris Hunter, who, in making these molecules, suddenly had an understanding of how two molecules stack on each other and realised what everybody else thought about how and why molecules stack on each other. Everybody else didn't really understand it, and he did. We published a wonderful paper in 1990. It's been cited 2,500 times. It got me into the Royal Society. It's now got him into the Royal Society. But you can see that I work in an area for some time. I get bored with it, and suddenly I see a new area that we can move into, and that's what keeps me young. Actually, is to keep moving into something new every few years. To be honest, I get bored after 5 or 10 years.
Well, I asked Martin Rees, Lord Rees, what was the secret of his success, and he said, a short attention span.
Yes, that's right. And I frequently have 2 or 3. So I have a research group. I frequently have 2 or 3 strands going. So the phone call from Addenbrooke's in 1977, we had some in pharmacy, led to a whole stream of doing spectroscopy on live cells. We ended up understanding how some molecules get metabolized inside cells. We ended up understanding something about the biophysics of biodegradable polymers. And then I realised that wasn't going anywhere. And so I keep moving fields, within the broad spectrum of chemistry, organic chemistry, inorganic chemistry, biological chemistry. I taught organic chemistry for 25 years. When I got a readership, as you know, you have to choose a title. Yeah. And I said to my head of department, I'd like to be reader in chemistry. And he was shocked, because everybody else had been reader in inorganic chemistry, or organic chemistry, physical chemistry. I said, no, I just want to be reader in chemistry. He said, okay. And then 1996, we had a chair of inorganic chemistry that we couldn't fill for a variety of reasons. I was by now a fellow of the Royal Society, had chair offers from elsewhere. I was still a reader. And David King was the head of the department. I said, look, you've got to make me a professor. And look at my molecules. My molecules are just as inorganic as organic, so they may be professor of inorganic chemistry. The inorganic chemistry community has never forgiven me, because I'm really, at heart, I'm an organic chemist. The organic chemistry community sees me as a traitor. The great advantage of this is that the Royal Society of Chemistry has left me alone, because it's still, until very recently, it's been tribal, organic chemists, inorganic physical. I'm not interested in that kind of tribalism. So I keep changing what I'm doing. I think it keeps me young. But to come back to your question about PhD students, my research group is usually about 50% postdocs. I've been head of the chemistry department. I was chair of the RE committee, national RE, and now head of the School of Physical Sciences. My technique, if you like, is to set up a laboratory, have very bright postdocs, very bright students, set a tone of being exploratory. Nothing is off limits. I've published a paper on spiders' webs. I've tried to use NMR to look at wet sand. I've done all kinds of strange things. Nothing to me is off limits by way of experiments. I had a PhD student who put black currants in a spectrometer to try and see what biology we could learn about black currants. I'm entirely scientifically promiscuous, if you like. As long as it's interesting, as long as it's new, as long as it's something where we stand a chance of learning something new, I'm happy for the group to do it. So there's an overall broad strategy, if you like. I mean, strategy is rather a grand word. There's a way of operating that I've got, whereby I have very bright young people. I expect them to talk to each other a lot, and to talk to me when I have time. And I say, why don't we suggest this? Or why don't we try that? But the best students do what I did, and what my best students have always done, which is say, that wasn't a very good idea, I've had a better idea. So Chris Hunter, who I mentioned before, my first FRS student, I gave him some results in, when did he start his PhD? In 1986. I gave him a paper I just published on how porphyrin molecules, the flat red component of hemoglobin, how these come together. And he said, came back to me a week later, he said, I've read that. He said, it's crap. Not the experiments are fine, the explanation, he said, it's all wrong. And I said, that's fine. You go away, you devise an experiment that distinguishes my explanation from what you consider to be the correct explanation. And that's what he did. And that's what led to this paper that made us both quite famous. So I'm very happy when my students come up with better ideas than I've got. My technique as head of chemistry was always to try and appoint people who are better than me. I mean, that seems to me the only way to. I'm very taken by Harry Truman's quotation that there's no limit to what you can achieve if you don't mind who gets the credit.
That's very nice. Just linking it to your current work, I read an account of your current project, which I don't know whether you can explain in simple words, but it seemed, I remember that something lodged in my memory after reading it, something about C60 and some way of, I mean, it seemed, why I'm asking today, as you know, the world leaders are meeting to discuss carbon emissions and problems of this, and as you've seen in the interview I did with Richard Friend, I was very struck by his suggestions of what might happen with polymers and light-emitting polymers and so on. Is any of your work at all related to problems of carbon capture, etc.?
Well, no, it isn't really. The main thing that we're doing is, during the 1980s, as I mentioned, we were making molecules to behave like enzymes, big molecules with cavities that could do chemical reactions. And what I realised by the end of the early 90s is that we and people all around the world invest a huge amount of time and money in building molecules that then don't work. Drug companies do it all the time. And they do that because we don't understand enough about molecular recognition, how one molecule recognises another. And I took my inspiration then from biology, the immune system. If you challenge a body with a foreign molecule, then your immune system has lots of different antibodies in it, and if there's one that is successful in recognising a foreign molecule, then it gets amplified. And what I've done is to devise a system, a chemical system, whereby you don't know what the successful molecule is going to be, but you have a a complicated mixture of molecules, then you add something that you would like to recognise, and the successful molecules are amplified at the expense of the unsuccessful molecules. And we call that dynamic combinatorial chemistry. And that's been pretty successful. One of the unexpected spin-outs from that 3 years ago was we discovered a system, and one of my postdocs, Dan Pantos, discovered a system that turns out to make tubes, nanotubes, and the nanotubes have a diameter which is exactly right to fit C60.
And C60 is?
C60 is a molecule discovered by Harry Kroto. It's one of the things that he got the Nobel Prize for. It's 60 carbons making a soccer ball, a football. And it happens to be an intriguing, rather beautiful molecule. It's not clear yet whether it has any importance in the world. It's just a pretty molecule. And it happens that it binds inside our nanotubes. And what we're now trying to do is to use our nanotubes as chemical reactors, because we've got very high concentration of C60 inside. Can you do chemistry on C60 inside these nanotubes? It's not directly related to anything to do with CO2 in the atmosphere and capture. And I do, sometimes I sit and I ask myself whether I feel guilty that everything I'm doing is really rather abstruse and pure, if you like. And should I be switching my research effort to solving some economic problem, carbon capture or conversion of light energy into electricity or something? And I know that strategically we need to do that, and as my role as head of school, I'm beginning to move the School of Physical Sciences to say we need collectively to be tackling these big societal problems. But I think that, especially as I don't spend more than 20 or 30% of my time on research now, I think it's too late for me to make that kind of a switch as a leader. I don't happen to be part of a big group, someone else leads. I'm not going to be able to change my style of research now. My style of research is essentially random, exploratory, and you never know what you're going to discover. It's quite cruel for PhD students. It's brutally Darwinian, my way of doing research, in that research students who do really well flourish, and if they're not lucky or they don't have the flair, then I don't have, if you like, a systematic research project. Let's do this and this and this, and we'll get a paper at the end of it. I've never worked in that sort of systematic way that would provide the safety net for a student who's unlucky. So I'm not good at doing strategic, directed research. It's just not my style. It's for others to judge whether I've wasted millions of pounds of public money over the last 40 years.
I've got 2 or 3 more questions. One is coming back to your own work. That you gave a particular date when suddenly your life changed, and there probably have been other moments. I'm always curious as to how people have their most formative ideas. I'm talking to distinguished scientists. Many of them find that it's when they're going for a walk with a friend and chatting, or having a bath, or listening to music, or random moments when suddenly things come together and they understand something afresh, a large breakthrough. Are there any patterns in your life in that way?
They don't tend to come that way. So the other big date that is etched on my mind is 17th September 1992, the day after the pound fell out of the European monetary system. You may remember Norman Lamont was Chancellor of the Exchequer. Interest rates went up to 15%, and then over a day, then the pound fell out and everything dropped. That week, the Royal Society of Chemistry had its annual congress in Dublin. And the day after that, on the Thursday, I was giving a lecture, and it was the first time I had lectured about doing chemistry inside these cavities that I've described. I had a wonderful student, Harry Anderson, who's now a professor at Oxford, who'd made this beautiful molecule, the cavity. And then the next PhD student, Chris Walter, had made two molecules which bind inside the cavity and react with each other. And it was something I had imagined, for 10 years, and it worked. And on that day in September 92 was the first time I gave a lecture on it. And in the audience, there were two people I greatly admire. One was Chris Hunter, my ex-student who I mentioned before, and the other was a man called Fraser Stoddart, now Sir Fraser Stoddart, who's a great pioneer of our kind of chemistry. I had lunch with them after my talk, and they said, yeah, that was great, that was interesting. But it's not a general solution to the problem of molecular recognition. One student had taken 3 years to make the cavity, another student had taken 3 years to make two components, and we were lucky it worked. Most times when people do that, it doesn't work. And as we talked over lunch, it became clear that there was something fundamentally wrong with the conventional chemist's way of making molecules, clicking things together in an irreversible way. See a good example. When you make Lego, when you make build, you can always take the Lego pieces apart and put them back together again in a different way. So you can proofread your Lego building until it's perfect. The conventional way that chemists make molecules is like if you ever made Airfix aeroplanes with glue irreversibly. And we realised during that conversation on 17th September that the solution, the general solution to the molecular recognition problem was to make big molecules in a way that mimics Lego reversibly. Okay? So you can proofread. And so if you have the molecule you want to recognise and you offer it this large library of different molecules, it will find the correct one and it won't find the incorrect ones. Now, once you've found the correct one, that stabilises a correct one. And if you now allow all the others to keep falling apart and remaking, then you can see that you will get more and more successful molecules at the expense of the unsuccessful molecules. That's what I call dynamic combinatorial chemistry. Somebody must like it. The Royal Society is giving me the Davy Medal this year for developing it.
Congratulations.
It's a very powerful idea, and I went home from that meeting really excited. By the end of the week, I'd sketched out the whole grant proposal. I knew what we needed to do. In intellectual terms, what we need to do, not in practical terms. And then I thought, why hasn't anybody else done it? And I still don't know the answer to why no one else has done that, because it's obvious. As soon as you start telling people about it, they say, oh yeah, well, that's obvious. Nobody had done it. Well, people had done experiments along those lines to solve particular problems, but what nobody had done, I did it, and independently, Jean-Marie Lame, who has the Nobel Prize for chemistry in 1987, came to the same conclusion. Jean-Marie and I both realised that this was a general solution to a problem and not just a way of solving this problem or that. So my best idea, that's the only good idea I've ever had in my career, that idea came out of a discussion. I'm not good at sitting in a darkened room or walking the hills with or without music and coming up with an idea. My ideas come from sitting and discussing with PhD students and postdocs. Maybe in the tea room, maybe in the pub, maybe in my office, I'm not aware. Maybe walking the hills, but in having a scientific conversation. And it's in the to and fro of the conversation that I get ideas. I don't come up with ideas.
Well, many of the scientists I've talked to, for example, Sydney talked a lot about his conversations with Crick, and Gabriel talked about conversations with Pat, and so on. It's often two people who get to know each other very well, and then they spark off each other. So
yes, and when I'm sitting with two of my postdocs, and I've got two very good postdocs right now, who've done some lovely work on these nanotubes and C60 and so on. And then when they write a paper, they write the draft paper, and they give me the draft paper. When I'm sitting and talking with them, what do we really mean in this sentence? And what do we really mean in that sentence? That's when actually a lot of the hard work of interpreting it and deciding what we've really discovered and what it really means, that's when it happens, is after the event. I'm not hypothesis driven in the way that now biology, conventional biology now, is very hypothesis driven. um, this enzyme, that coenzyme, this factor, let's draw up a hypothesis and then go out and test it. I don't work like that.
Mm.
I like to look at results, and in the words of Laurie Hall, who I don't know if you ever came here, Laurie Hall, I did a sabbatical with when he was at Vancouver. He came here to Addenbrooke's a few years ago, died last month. Laurie would always say, listen to what the science is telling you. And that's what I do. I look at the results and I listen, and I look at what they're telling me and I listen to what they're telling me. Preferably hear things that other people have missed.
That's a very nice account. And the last question I had is, you said you only spent 25% of your time now on research, and another 75% is running the School of Physical Sciences, no doubt. Thinking about the structure of the university and ways in which this university can protect good science of the idiosyncratic kind that you have been practicing yourself. Do you think, well, firstly, is Cambridge a good place for this kind of work? And secondly, I suppose, any thoughts you have on how it can be protected and encouraged?
I think it's problematic. And Cambridge has been very good for quirky individuals. There were some who were not necessarily well organised, but who were protected by living in college or the college environment. When I first came here as a junior demonstrator in the early 70s, whatever I needed, I could go to my head of department and say, can I have? And he would say, yes, no problem. Head of department now don't have that resource. Nobody asked me when I was appointed what science I was going to do, how I was going to fund it, what was my strategic plan for the next 10 years. Nobody asked me that. Now, of course, this is going to sound immodest, not all the appointments that were made in that way turned out very successfully.
Mm.
But actually quite a lot of them did, at least in chemistry. So I think what was going on then was they were somehow picking talent and giving talent some freedom and some resource. It's increasingly difficult for us to give resource, significant resource. Very talented people now are in great demand and can demand huge startups. We're in competition with American universities that give startup packages of the order of $1 million routinely to chemists at the beginning of their career. If we can't compete with that, then we will lose out. But if we do compete with it, we then get a set of people who are very determined, very focused, very ambitious. They're very good for themselves. They're not necessarily ideal for a community. And although Cambridge has often been a rag bag of quirky, assorted individuals, there was also a sense of community when I started in the chemistry department in the early 70s that I don't see anymore. It's not a problem that's unique to Cambridge, it's everywhere. That if you appoint people who are good enough to go out and get $1 million grants or £1 million grants in their first few months, they are usually people who are very focused, who have strategies, who know what they want to achieve in a way that I never did, I never knew I wanted to achieve. It's hard to manage. People like that are actually quite difficult to manage if they don't have that sense of community. You ask them to do something to the department or the university or the college, they will explicitly or implicitly make a calculation of whether an hour spent that way is an hour of their time well spent or not. And that's going to make it particularly difficult in future for colleges in science. Because colleges don't if you like, pay anything like the going rate, either in monetary compulsory or undertone. So I think it's very, very difficult. In some ways, we're better placed than other institutions. If we can get in big grants, if we can all give up a little bit of our individuality for, if you like, the common good, to get in a big grant to look at sustainability or to develop new materials for photovoltaics or for batteries. If you have a big grant that covers 10 people, then within the protective envelope of those 10 people, you can afford to have individuals who are not necessarily team players, who don't follow the strategic plan, but follow their own creative noses. So it may be that by putting people together into big consortia, you've got enough space to protect the individuals that really do the creative things, but who don't necessarily flourish in our present sort of target-oriented society.
That's a good answer.
That's an optimistic answer.
Well, let's see if you can achieve it. Always in these interviews, there's so much that one has to leave out: your wife, your family, your hobbies, your everything else. But is there anything really important, a question that you would have liked me to have asked you, which I haven't touched on?
To me, it's been very important to have a happy and stable home life. So I can concentrate, if you like. I've been very involved in bringing up my children. I often used to go home at 3:30, 4 o'clock in the afternoon, collect the children from school, take them to swimming, trumpet, piano, all of that, and then work again in the evening.
What children do you have? I mean, boys, girls?
So we have a son who's now 32, who's an assistant professor of physics in Mexico. He married a Mexican. Our daughter is 30 and she's the academic administrator at Somerville in Oxford. And my wife's a biologist, although she's not currently working. So all of that dimension is pretty important to me. The only thing I do around the house is cook. I do the shopping, I do the cooking.
That's quite a contribution.
Well, I have no idea what happens inside the car. My only contribution to decorating the garden is to write the cheque.
Well, it sounds as if you've had a pretty fulfilled and happy life, and I wish you all success with this new task with the school, and it's been a great pleasure to interview you.
Oh, well, thank you.
Thank you very much
Great pleasure
indeed.
to talk about it.